The next question tells us, what is the next question? Which of the following hormone level will cause releasing the ovulation? Shall I go to the previous slide? Once I already explained this one in the previous slide, so I'm going to the previous slide. Look at here in the previous slide, ovulation. Under the influence of what ovulation takes place? Under the influence of what? Graffian follicle, matured Graffian follicle rupture and release the secondary oocyte and ovum. We have seen LH, estrogen, but in the question LH is not given, student. Look at the question. The question tells us under the which hormone influence the ovulation takes place. Look at the option. High concentration of estrogen. Absolutely correct. There are two hormones are affecting, LH, pituitary hormone, and also estrogen hormone. But in the question, high concentration of estrogen only given. High concentration of progesterone, no sir. High concentration of progesterone maintain the endometrium thickness. You got my point. Next, low concentration of LH. No sir, absolutely wrong. High concentration of LH only. You got my point. But that is not given. Now, the next hormone is given. What is that? High concentration of estrogen. So, our exact answer is high concentration of estrogen. Last option, low concentration of FSH. Low concentration of FSH is not influence the Graffian follicle. So the right answer is under the influence of two hormone, matured Graffian follicle rupture and release the secondary oocyte. This process called ovulation. So we can easily tell like this: ovulation is controlled by pituitary hormone LH, ovarian hormone estrogen. So our answer is option A. Shall we move on to the next question, student? Look at the next question. Chemistry, sir. Waiting. Students are waiting.
Yes, okay, dear all. Are you ready? Okay, aniline, there is NH2. Aniline is reacted with the bromine water, right? What is bromine water? We can represent as Br2 water. Dear all, aniline is a very strong ortho-para directing group and bromine water is a very strong brominating agent. So what happened? All the ortho and para positions are occupied by, this is ortho and para, they are occupied by what? Bromine. You know, it's a white colored precipitate, 2,4,6 tribromoaniline. Done. And the resulting product is treated with aqueous solution of sodium nitrite. This compound is treating with what? Aqueous solution of sodium nitrite. What do you mean by sodium nitrite? NaNO2, sodium nitrite in presence of what? HCl, dear all of you. What is this reaction, all of you? NaNO2 and HCl, diazotization reaction. During diazotization, what happened to the NH2 group? It becomes N2 plus Cl minus with all the ortho-para positions by bromine. Next. And after that, this compound's form is converted into tetrafluoroborate, which is subsequently heated. What? This compound is converted to what? Tetrafluoroborate. Dear all of you, if you want to get tetrafluoroborate, what should be treated? You should treat with HbF4. What is HbF4? Is called the fluoroboric acid. HbF4 having two ions, dear all, H plus and BF4 minus. This H plus Cl minus removing as HCl, this BF4 come instead of Cl. So you will get the compound benzene with N2 plus BF4 minus with bromine, bromine and bromine. What is its name? Here, second, fourth, sixth position, we have bromine and it's called benzene diazonium tetrafluoroborate. 4 fluorine, right? Tetrafluoroborate. And this compound is heated, dear all of you. Once you heat this tetrafluoroborate from BF4, you add 1 fluorine on benzene ring directly. From BF4, take 1 fluorine and add on benzene ring. You will get this compound. Dear all, this is the product. What is the byproduct? From BF4, 1 fluorine you added on benzene, right? You got this one. So one BF4, you took only 1 fluorine. So how many fluorine left? 3 fluorine. So that means BF3 is one byproduct and also this nitrogen. So the major product is which one? This one. 2,4,6 tribromo fluorobenzene. What is that? 2,4,6-tribromofluorobenzene. Dear all of you, the question from benzene diazonium chloride. You know, benzene diazonium chloride is a very important compound from which we can prepare a lot of chemical compound, like especially chlorobenzene, bromobenzene, iodobenzene, fluorobenzene, benzene, phenol, anything, right? So it's very important compound to know what are the applications of diazonium salt and their preparation. Hope you clear the question. Here the option is number 4, 2,4,6-tribromofluorobenzene. Yes, Sandeep sir, let's go to the next biology question.
Right, thank you, Deepak sir. I hope students are enjoying this session. Shall we discuss the next question? The option is D. Now I'm also learning chemistry. Now what is the answer? D is the right answer. Look at the next question. The next question tells how do ovulated oocyte gets into a uterus. Ovulated oocyte gets into uterus. Keep in mind, all of you, male female gametes, the male gamete, sperm, have a long tail for the movement, but secondary oocyte don't have any tail, don't have any locomotory organ to move from here to there. With which parts it will be moved towards the uterus and it will enter into the fallopian tube? We know, we have already discussed. Let me read the question. Let me read the option. Because ovaries are within the uterus. Ovaries are within the uterus? No sir, it's not correct. Look at here. I'll tell you. This is fallopian tube. This is fallopian tube. Now we know, we have already discussed. Now this is uterus. And this structure is endometrium membrane. This is cervix, narrow cavity, and below the cervix, vagina. We have already discussed this one. Look at here. This structure is called finger-like projections, fimbriae of ovary, fimbriae of fallopian tube. This is ovary. From the ovary, what release? Secondary oocyte release after ovulation. This ovulated secondary oocyte received by finger-like projection and put into the, that means they receive into the infundibulum. My dear students, keep in mind, fallopian tube of female is made up of ciliated epithelium, ciliated columnar epithelium. So here there are cilias are present. The movement of this cilia movement helps to move this secondary oocyte towards the ampulla isthmus junction. So what is in the wall of the fallopian tube? Small cilia, ciliated columnar epithelium. So let me write here, these structures are known as ciliated columnar epithelium, helps to move the egg towards the uterus. You got my point? Now pick up the correct answer. What is the right answer now? Cilia within the oviduct create the current that sweep the egg toward the uterus. This option is absolutely correct. Look at the next one. The oocyte actively swim. What is the organ helps to swim? Nothing is there. No cilia, no flagella, nothing is there in the secondary oocyte. So oocyte, secondary oocyte independently cannot move. So what are helping to move the secondary oocyte? Ciliated columnar epithelium of fallopian tube. So our answer is option 2. I hope you understand. Shall we give to the Deepak sir? Look at the next question. Next question is chemistry. Now I'm inviting Deepak sir.
Yes, Sandeep sir. Dear all, next question from amines. Which of the following statements about primary amines is false? As I told you what is primary amine before, that is primary amines means which having what? NH2 group, right? So question number 1. Alkyl amines, okay, alkyl amine, for example, let me take it methylamine, right? It's the alkyl group, right? Methylamine. This alkyl amines, they are saying that it is a stronger base than ammonia. Are you agreeing with this statement, dear all? You know, alkyl amines are more basic than ammonia. Yes, it's correct. Because alkyl amines having which group? Electron donating group. Electron donating group, if it is there, it will enrich the amount of the lone pair. So it's ready to donate lone pair. Therefore, they are Lewis base. So electron donating groups are there in alkyl amine. It will increase the basic character. Yes, the first statement is correct. But our question, which is the false statement. So not option number 1. Alkyl amines are stronger bases than aryl amine. Yes, of course, dear all of you. This is the order. Alkyl amines are more basic than ammonia. Ammonia is more basic than generally which one? Aryl amine, for example, aniline. You know the aniline, the lone pair is involved in resonance, so it is not available for donation. Same like our amide case, what we discussed before. So the aromatic amines are also less basic and alkyl amines are more basic. Yes, second statement also correct, but that is not our answer. Option number 3, alkyl amines react with the nitrous acid to produce alcohol. Is it? Let me take one alkyl amine, for example, CH3NH2. What do you mean by diazotization? Treated with the NaNO2 and HCl at 0 to 5 degrees Celsius. During this reaction, what happened to the NH2 group, dear all? It converted to N2 plus Cl minus, diazonium salt. You know that here resonance is not possible. In alkyl diazonium salt, the resonance is not possible, therefore it is highly unstable. Since it is highly unstable, it immediately reacts with the water and it will give you alcohol, dear all. So alkyl diazonium salt is highly unstable, so it won't stay for a long time. It unstable, it immediately reacts with the water and will give you what? Alcohol. So that is a correct statement. Then what about the last one? Aryl amine, for example, that is aniline. Aryl amines react with the nitrous acid. Same reaction. Nitrous acid means what, dear all of you? HNO2. That is same. Dear all, NaNO2 and HCl when react, you will get NaCl and HNO2, right? So both are same, okay? Nitrous acid, diazonium. So NH2 group converted to what? N2 plus Cl minus. It immediately don't react with the water because it is stable. You know, it is resonance stabilized compound. So immediately it won't react with the water. Only by warm condition, heating, then only it will react with the water and it will give you phenol, benzene with OH. But aryl amines react with the nitrous acid to produce phenol. Yes, it can produce, but only by heating. So among this given 4 option, the only one incorrect statement is which one? The 4th one. Clear all of you? So let's go to the next question, biology. Sandeep sir, please.
Right. Look at the next question, my dear student. What is the next question? Capacitation occurs in. What is this capacitation? Capacitation is nothing but that functional movement or functional change in the sperm. This process takes place in the vagina or female reproductive tract. Once ejaculated, sperm ejaculated, ejaculation done, that means semen deposited in the female reproductive tract. Look at here, let me take the sperm. Look at here, this is the sperm head part. This is sperm head part. We know sperm has neck part, head part. Let me draw properly. This is sperm head part. Acrosome is there, head part is there, middle piece is there, and also tail also there. Yes, we have seen this one. This is the sperm structure. Now what happened inside this head part? Haploid nucleus is there. Yes, my dear students, on the top of the head part there is modified Golgi. What is the structure called? Acrosome is present. This is essential for the penetration into the egg. Penetration into the penetration of a sperm into the egg. It is essential. So what is this part? Tell me. Acrosome. And what is this part? Head part. You got my point. Head part, and this is haploid nucleus. Haploid nucleus. Am I clear? Here there are mitochondria are present. We have already seen mitochondria are there. What is the meaning of capacitation? Once sperms are deposited in the female genital tract, there are on the top of this acrosome, there are small glycoprotein molecules are there. Look at here, I am representing glycoprotein molecules are present like this on the top of the sperm. These molecules are glycoprotein molecules. Am I clear, students? Now what happened? These glycoprotein molecules will be removed by the vaginal fluids. Once this ejaculation done, so whatever the fluids are there in the vagina, that fluids remove this glycoprotein on the surface of acrosome. So when these glycoproteins are removed, when these glycoproteins remove what happened? Calcium ions from semen influx into the sperm. Calcium ions from outside. The semen contain calcium ions also there, right? We have seen the seminal vesicles are secreting that seminal plasma in which calcium ions are one of the part. So these calcium ions are influx entered into the sperm and the sperm actively swim towards the egg. That means calcium ions are helps in the activation of sperm. So this complete changes and functional changes in the sperm called capacitation. So this capacitation process takes place in where? Female reproductive tract. So our answer is C. Keep in mind, capacitation belongs to sperm. You got my point? This is not takes place in testes. This is not takes place in vas deferens and epididymis. This process takes place in female genital tract. So the answer is option C. Shall we go to the next question? Next question. Chalo. Chemistry sir.
Yeah. Dear all, the question is, which of the following gives a soluble complex on reaction with benzene sulphonyl chloride, right? What you mean by benzene sulphonyl chloride? You know, it's called what? Hinsberg reagent. What is the structure of benzene sulphonyl chloride, dear all? This is called benzene, this is called sulphonyl, and it's called chloride. Benzene sulphonyl chloride is the compound called what? Hinsberg reagent. You know what is the purpose of Hinsberg reagent? Hinsberg reagent used for the distinguishing of what? Primary, secondary and tertiary amines. What is the observation you have studied? Primary amines, when reacted with the Hinsberg reagent, you will get a precipitate. It is soluble in alkali. That is the question. When this compound treated with this benzene sulphonyl chloride, it is soluble. So dear all, they are asking you which is a primary amine. Very simple. And what about secondary amine? Secondary amine, when you treat it with a Hinsberg reagent, you will get the precipitate, but it is insoluble in alkali. What about tertiary amine? It won't react. Yes. So let me take this compound. The question is, what is a primary amine, or which is a primary amine in this compound? Which is a primary amine, dear all? Option number 1, because it is having NH2 group. It is primary. NH group means what? Secondary. Alcohol won't give this test. And N means what? Tertiary. So primary amine and tertiary amine, secondary amine can be distinguished by the method called what? Hinsberg reagent test. Hinsberg reagent is what? Benzene sulphonyl chloride. Once it reacts with the primary amine, you will get a compound that is soluble in alkali, whereas secondary amine also will react, but it is insoluble in alkali. Tertiary amine won't react. So this is a distinguishing test for primary, secondary, tertiary alcohol Hinsberg test. Yes. Next question, biology. Sir, please.
Now look at the question. This question is from implantation. Yes or no? Look at this question. Identify the human developmental stage showing as well as the related right place of its occurrence in the normal pregnant woman. One thing keep in mind, student, whenever in the diagram, if you are identifying the cavity, look at here, this is the cavity. Let me write here, this is trophoblast cell. The smaller outermost flattened membrane cells are called what? Tell me. Trophoblast cells. Am I clear? Now what happened? Inside this one, inner cell mass is there. Let me represent this one. This is inner cell mass. So what is formed here? Cavity. This cavity is called blastocele. Keep it in mind. Blastocele. When a cavity is formed, it's look like a cyst. So the stage of this embryo called blastocyst stage. Blastocyst stage. What is this cells called? Trophoblast cells. Trophoblast cell. What is this cells called? Inner cell mass. Inner cell mass. One more thing keep in mind, student. This trophoblast cells release the enzyme called trypsin enzyme, which dissolve the zona pellucida, so that after fertilization, exactly this stage of embryo come and implant to the endometrium. endometrium of uterus. This process is known as, tell me, implantation. Implantation. Am I clear, student? Implantation. So at which stage the embryo is come and implanted into the uterus? Blastocyst stage. So look at the question. The question tells us, identify the human developmental stage shown in. Sir, the cavity is there. This cavity is called what? Tell me. Blastocele. Blastocele. Now here we have seen this one. What is this? Blastocele. So whenever the blastocele is there, that structure is called the blastocyst stage. Pick up the correct answer. Look at here, blastocyst is there. But I told you, when blastocyst is formed, the right place of attachment is where? Endometrium of uterus. So what's the correct answer here? Blastocyst, uterine wall. After 7 days of fertilization, this blastocyst stage come and attach to the endometrium membrane of uterus. This process called implantation. Which hormone is essential for this implantation and making that endometrium thick? Progesterone hormone. So what is our correct answer? This is not a late morula. If it is a late morula, we would have not identified what? That blastocele. Blastocele is formed nothing but it is a blastocyst stage. So our correct answer is option C. Or correct answer is option C. The cyst is blastocyst, the stage is blastocyst, and the implantation site is endometrium membrane of uterus. So the next question comes, sir.
Yes, Sandeep. Dear all, let's go to the question. Which of the following can give a carbylamine test? So what's the question? What do you mean by carbylamine test? Dear all, carbylamine test is also called a what? Isocyanide test. You will get a foul smell, right? It is a test is given by, this test is only given by primary aliphatic and primary aromatic amine. Keep in mind. Carbylamine test is also called isocyanide test. It is only given by primary aliphatic and aromatic amines. What is the test? For example, let me take a primary aliphatic amine. It is treated with the what? Chloroform. You know what is chloroform? CHCl3 in presence of a base called KOH. Then we will heat. So what are the reagents in carbylamine test? That is a primary amine and the chloroform, KOH, then you heat. The primary amine converted to RNC. RNH2 converted to RNC. What is the functional group called? NC, isocyanide, right? Therefore, this test is called isocyanide test. It's having a foul smell. What are the byproducts, dear all? KCl will get it along with the removal of water. Okay, so this is called isocyanide test. So keep in mind, isocyanide test is also called what? The carbylamine test. Carbylamine test is only given by primary aliphatic and aromatic amine. What is the test? The amine you treat with the chloroform and KOH or any base, and on you heated, you will get the amine converted to isocyanide. Then along with the KCl and water. So what is the compound answer? Which of the following will give carbylamine test? Which is give carbylamine test? That is our amine. Which amine? Primary amine. What about this amine? It is tertiary, right? What about the second option? That is a secondary amine. What is the last one? That is an amide. Okay, starting material for or starting reagent for this carbylamine test is what? Primary aliphatic or primary aromatic amines. Clear the question, all of you? Yes, let's go to the last biology question. Sandeep sir, please.
Thank you. Look at the next question. I hope you all are enjoying, right? The next question, in telolecithal egg, the yolk is found. Sir, what is this telolecithal, centrolecithal, mesolecithal, hololecithal? All of you keep in mind and write down in your notes types of eggs based on the yolk distribution. Now, I'm going to explain types of eggs. Now here, types of egg. Based on what? Tell me. Based on yolk distribution. Yolk distribution. Okay. Keep in mind. I will give you the example also. Okay. First one, hololecithal. First one we can write hololecithal. Or we can write homolecithal. Homolecithal. homolecithal egg. What exactly the meaning of this homolecithal egg? If you take this is egg, okay, the yolk content is equally distributed all over the egg. So this is cytoplasm, this is nucleus, and I'm representing with this green color. The yolk is equally distributed all over the egg. Am I clear? Now I'm writing here yolk distribute equal in egg. Am I clear? It equally distributed all over the egg. We can write equally distributed all over. Such type of eggs are called homolecithal. Example, human egg. Example, human egg. I hope you get my point. Number 2, second one. Second type of eggs are called telolecithal egg. To understand this telolecithal eggs, just assume a chick egg. When we boil the egg, we identify, if I draw the diagram, look at here, this is egg, this is chick egg. We identify the yolk is one side only. This is total calcium carbonate and the white color part is there, which is the glycoprotein. And where is the yolk? Yolk is one side of the egg. This is yolk. If yolk is one side distributed in the egg, such type of eggs are called telolecithal egg. Example is bird eggs. Example is bird eggs. So I hope you get the answer. What is the answer here? In telolecithal eggs, the yolk is found one side. So answer is B. The right answer is what? B. So what is the third type of egg? Let me finish this one. It will be useful for you. So the third type of egg is called centrolecithal. Centrolecithal. That means the name tells us the yolk is majorly accumulated and located in the center of the egg. So if I take, this is the egg, this is egg, okay, and where is the yolk deposition takes place? The major amount of yolk distributed or accumulated in the middle. So this is yolk. Such type of eggs are called centrolecithal eggs. So what is the example of centrolecithal? Keep in mind, in many times competitive purpose, they are asking the centrolecithal egg example. Insect eggs, cockroach, or whatever the insect you take. So insect eggs. Insect eggs are shows what type of centrolecithal. So based on the yolk distribution, there are three type of eggs: centrolecithal, telolecithal, homolecithal. Human being eggs are homolecithal. The amount of yolk is majorly, the amount of yolk is the pattern, the cleavage pattern is depending on this what? Amount of yolk also. If the amount of yolk is more, there is a meroblastic cleavage. The amount of yolk is less, that is holoblastic cleavage. That we will discuss in the cleavage pattern topic. Okay. So the right answer of the question is option B, that is one side of the egg. Okay. Shall we go to the next question? Deepak sir, come.
Yes, dear all, this is the last question in this series. We have a chemistry question. Nitration of aniline in strong acidic medium also gives m-nitroaniline because you know that during aniline nitration, aniline means benzene with NH2. Once you treated what is nitrating mixture, concentrated HNO3 and concentrated H2SO4. Right, so nitro group are coming to the system and it will goes to ortho and para position because aniline is an ortho para directing group. So normally we will get total, there are three products, right? NH2, one nitro group go to para position. You will get one another NH2, one nitro group goes to meta position, and you will get another NH2 and NO2 group to ortho position. This is what para nitroaniline, meta nitroaniline, ortho nitroaniline. So I think we know NH2 is a ortho para directing group. Then why this NO2 groups to meta? There is a confusion, right? You know, para is always the major, around 51% and 47% meta, remaining 2% ortho. So the question, why there is a substantial amount of meta product form during the nitration of aniline? That is the question. So dear all, what is the reason? We know that aniline is NH2, it is a ortho para directing group. Here you are treating this aniline with what? Concentrated HNO3 and concentrated H2SO4. It's a very strong acidic medium. What is present in acidic medium? H plus ion. H plus is acid. NH2 is base. What happened? Nitrogen having a lone pair. By donating this lone pair, it will take this proton. So what happened to this NH2 group? It becomes NH3 plus. Dear all, aniline is an ortho-para directing group. It's a base. When react with acid H plus, it becomes N-benzene with NH3 plus. It is known as anilinium ion. This is aniline. On acidification, it's become anilinium ion. Aniline is ortho-para directing group, whereas anilinium ion is a meta-directing group. So because of the formation of anilinium ion, it's giving around 47% what? Meta product. So what is your answer, dear all? I think option 4 is the correct answer. In acidic medium, aniline is present as anilinium ion. So because of the formation of anilinium ion, we are getting a substantial amount of what? The 47% meta product. Hope you're clear. Sandeep sir, please.
Right.
So we completed around 30 questions from the series, the first series of the chapter. The chemistry we discussed fully, that amines chapter, previous year question. So which all chapters we discussed in biology?
Right. Deepak, today we discuss sexual reproduction in flowering plant. That is the second chapter from the 12th grade, and also human reproduction.
All right.
So this series will be continue, and we will discuss more questions from the different chapters.
Yeah.
So students, whoever seriously preparing for the NEET like competitive purpose, kindly watch this video and boost your content. And shortly a physics are also going to join with us. So we will make a 3 subjects parallel simultaneously, 3 subjects, 3 questions we will discuss.
Yeah. So make more concentration on your subject and keep practicing the question. We will be available with all other chapters also. So see you soon. Dear all, thank you all.
Thank you very much.
Brilliant Qatar, your trusted coaching partner for IIT, JEE, NEET, Science and Commerce tuitions with 10 years of excellence in quality training. Brilliant Qatar.